Got it. Yeah, thank you so much, Hannah, for the introduction and also having me. It's my first time in Atlanta, and so far so good. It's, thank you all for the hospitality and coming to the talk. Is everyone on Zoom can hear me okay? Can everyone? Yeah, all right. So one of the most fascinating things about the brain to me is its ability to interact with the environment in a flexible way. You know, it's you can think of the brain as an input-output machine. It interprets the environment through the sensory system and acts on the environment through the motor system. But this relationship and interaction with the environment is highly dynamic and flexible. And how does the brain, how do the brain circuits subserve this flexible interaction with the environment? That's really the core interest that's driving the research in my lab over the last 12 years now at UCSD. And with this point of view, we work on a number of different topics. Here I'm just listing the three main thrusts of the lab and the topics that my lab pursues: motor learning, decision making, and olfactory learning, olfactory perceptual learning. I'm happy to talk about any of them, but for a talk, I have to pick one. And I wanted to share with you some of our latest developments looking at brain mechanisms of motor learning, okay? And yeah, please stop me to ask questions anytime. I'd much rather have this be interactive and understandable than just going through all the slides. So yeah, please stop me. All right, what is motor learning that we study? Well, it's you know, different. There are different types of motor learning. Different people define it differently. A particular type that we are interested in is the learning of motor skills. And motor skills are learned, reproducible movements that reliably achieve behavioral goals. And this reliability, to me, is really the key to what's being learned. So, you know, just imagine you're learning to play tennis. And when you're a beginner, you might be able to hit a really nice shot once in a while, but it takes sometimes years of practice to be able to do that reliably across trials, right? So something in the brain is changing to allow you to do the same thing reliably, reproduce the same movements. So how does that brain do that? That's the key question that we've been after. Here's one other slide that I used to highlight this topic. So here's two videos of my first daughter. On the left is the moment that she walked for the first time in her life. And then on the right is 1 year later. And these are all actually this is 5 years later. So now she's much more capable at performing more complex locomotive movements. Two things I want to do on this slide. One is to really showcase how beautiful my daughter is. But the other thing that's perhaps more relevant to my talk here is that, you know, even the simple or common behaviors like locomotion are learned and practiced through trial and error, so that your movement becomes more reproducible and reliable across time. Okay, so that's really something. That's really what we want to understand in this line of research. So when we started this investigation a number of years ago, we started by trying to adopt this question in lab mice. And here's one of the tasks that we used to study this. So here the mouse is head fixed and given access to a lever that they can use to grab onto and move down, all right? And here, this is the first day that the mouse is performing this task. The gray trace is showing the movement kinematics on individual trials. So those are how the lever is moving over time, overlaid across trials. And you can see that you know, there's really no discernible pattern that's reproducible across trials. But through daily training over 2 weeks, this movement becomes more and more reproducible and reliable, so that this reproducible movement pattern emerges as a function of daily training. Okay, so hopefully you can see the analog to what I talked about earlier. Let's say a tennis swing. Initially, your swings are variable, but you develop a more consistent movement pattern, right? So we think we are tapping onto an important key aspect of motor learning that we're interested in. And I'm not going to go into all the things that we did in the past, but one of the things that we showed, I think, conclusively is that this initial phase of learning requires the primary motor cortex and its activity. Okay. And so a lot of our study focuses on changes in the primary motor cortex, or M1, during this initial phase of learning. A quick side note, yeah, please.
Yeah, so is that motor learning or reward
learning? Yeah, so that is an interesting question. This is definitely reinforcement learning in the sense that they're making this movement to receive a reward. But the fact that they're using different types of movements early on that all lead to a reward, but eventually by repeated practice, they just pick one of those movements and stick to it. That's the part of motor learning that we're interested in. Yeah. And yeah, great question, thank you. And so a quick side note is that we've shown also that if we train mice with this task for additional month or two, then this movement eventually becomes independent of M1. So mice can do this task without M1 when it's highly overtrained. But we are interested in the period, early period during which the behavior critically depends on M1, okay? And so one of the first experiments we did was to apply 2-photon calcium imaging. This is a technique that we often use to record the activity of a population of neurons. And one of the key advantages of this technique is that we can use it to record the activity of a population of neurons one day, put the mouse back in their home cage, take them out the next day, and very reliably identify and record from the exact same population of neurons across time for a number of days to weeks. And so we use this technique to study how the identified population of neurons, their activity might change during learning of this task. This is something we did a number of years ago, and I won't go into too much detail, but just to show you the key findings here. So here, this is the activity pattern of a population of neurons during the beginner phase of learning. Here, different colors correspond to the activity timing of different neurons. And each dot represents a trial at which the neuron was active. And hopefully you can see that at this phase of learning, the activity timing is highly variable from trial to trial. But with daily training over 2 weeks, each neuron begins to pick a favorite time to be active. And they are reliably active across trials at the same time relative to the movement onset, so that the whole population forms a reproducible sequence of activity. All right. And as a result, the population activity becomes similar across trials, which is quantified as a population activity correlation here that goes up as a function of training. And one of the key aspects of this is shown here. So as a result, the learned movement, this expert movement, and we'll come back to this later, but this learned movement is paired with a reproducible sequence of activity. But as I said earlier in the tennis swing, even the beginner can perform generate the nice shot once in a while, a nice swing once in a while. And we see the same thing in mice as well. Even in the early phase of learning, there are a small number of trials where the mouse generates this learned activity pattern, learned movement pattern, excuse me. But when we look at those trials, the activity pattern is very different, okay? So what this suggests to me is that, so there's a target movement that is being learned, and there are different population activity in M1 that can eventually generate the movement. And during learning, they are trying out different activity patterns until one particular activity pattern is somehow chosen, and something in the brain changes to be able to reproduce that activity pattern so that it is reliably paired with the movement that is being learned. Okay. So there's a degeneracy to the activity pattern that is paired with the target movement. And what is happening during learning is some changes that allow the brain to reproduce one of those activity patterns so that it is reproducibly paired with the learned movement. All right, so that's sort of the quick summary or background for the two stories that I wanted to share with you today. Two investigations at very different levels. So first, we're interested in the mechanism of the activity pattern changes, and we look at it at the synaptic level. Okay, so specificity of synaptic changes in M1 during motor learning. And in the second part of my talk, I will share with you the ongoing investigation looking at the role of this activity pattern. So I'll start with the synaptic level changes. And just to reiterate, this is really trying to understand how the population of neurons go from here to here. So very variable activation to reliable activation, okay? And we think synaptic changes are key to this observed change. And we started this by applying high-resolution imaging. This is a structural imaging of Z-stack of a sparsely labeled M1 neurons. We're looking at the layer 1, superficial layer, looking at the dendrites. And while you see these little protrusions out of the dendrites, those are dendritic spines or postsynaptic sites of these synapses that these neurons are receiving. And we showed that during the learning of this task, we can identify these synapses that formed during learning. So they didn't exist at the beginning of learning, but they formed during learning after a few days. And also there are some synapses or spines that existed at the beginning of learning, but eventually get eliminated during learning. So there is a turnover of synapses going into these neurons. Showing this in the schematic, we find that this distal apical dendrites on the superficial layer, layer 1, generates new synapses during learning and eliminates some of the old synapses. So there's a compartment-specific turnover of synapses onto these M1 neurons. And so this was, again, a number of years ago. And it's nice in the sense that, okay, so new synapses form as a function of learning, but it doesn't really tell us how these synapses contribute to the learned changes. And yeah.
Just a quick question. Is there a spontaneous change in synapses not during a learning task? So if you look outside the learning window, do you also see this or not happen?
You see some of this, but learning enhances that. And this is especially during the first 5 days of the 2-week-long training, this process is happening very fast. Yeah. Mm-hmm. All right. So yeah, back then, we didn't know what these new synapses did, right? We just knew that happened, but we didn't really know what these new synapses encoded in terms of information or activity. And a new approach became available in the last few years, mainly driven by the improvements of glutamate sensors called GluSniffer, originally generated by Roger Chen at UCSD, improved by Lauren Luger-Janelia, who is now also at UCSD. But now we can express this sensor on the dendrites and report the presynaptic release of glutamate. So we can image them at individual spine level when those spines are receiving the presynaptic release of glutamate. Okay, so basically when the presynaptic neuron is active, they release glutamate, and we can image that postsynaptically. And you see that the different synapses are active at different times, although they belong to the same dendrite. What did I do with my water? Oh, there it is. So taking use of this new sensor, we express this in M1 layer 2-3 neurons, and let the mouse learn this task and image the same dendrites across the learning period, and identify the new synapses that formed during learning, and looked at what kind of information they encoded. Basically, when are they receiving inputs? And here is 12 examples of new spines that formed while the mice were learning this task. And the activity of each of those spines is aligned to the movement onset shown in the dotted line here. And we were excited to find that many of these new synapses were active, time locked to when the mouse is performing this task, okay, when the mouse is generating this learned movement. There's a population of those, all of those new synapses. Many of the synapses are active right at the movement onset, or some of them before the movement onset, consistent with the idea that these synapses become active that eventually lead to the generation of the learned movement. Okay. And at a first glance, the activity of these synapses look a lot like some of the pre-existing synapses. And we call them movement-related spines. So these are spines that show increased activity, time locked to the learned movement. And these are 12 example pre-existing movement-related spines. And you can see qualitatively that the new spine activity is very similar to the activity of movement-related synapses or spines. So this is good. This is, to the best of our knowledge, this is the first time to show that the learning induced synaptogenesis leads to synapses that encode learning-related information. So that might have been a prediction, but it had never been shown, and it's nice to confirm that. And I think I saw a hand. Yeah.
Is there any spatial structure to the division of the spines that encode this information? They tend to be, for example, clustered together, or are they kind of random?
Yeah, that's really the key topic of the rest of this part of the talk. So yeah, a great question. Yeah, hold that thought. I think we can go to the model, and then if you have additional questions, we can come back to that. Yeah, thank you. Any other questions? All right. So okay, so new synapses encode learning-related information. That's good. Maybe not so surprising. What was very exciting to us is sort of schematized here, illustrated here. So here I am overlaying the activity of two spines. All right, the blue in new spines. Blue is a new spine, and red is a nearby movement-related spine. So their activity is different, but quite often they coincide. They're co-active. They're two different synapses next to each other that receive inputs at the same time. Okay. And this is more likely when those two synapses are close together. So new spines show correlated activity with nearby movement-related spines, especially when they are close together within 5 or 10 microns. So this is a very small scale coordination of activity that is happening on the dendritic branch. And for those of you that don't think about dendritic integration all the time, this is an exciting finding because previous work, mostly in slice, have shown that the location of inputs matters. Okay, so when multiple synapses near each other are co-active, they can have a supralinear cooperation to drive the postsynaptic neuron strongly. So even the same amount of input, shown in 3 arrows here, if they are distant from each other, they only add linearly, and they only have a small effect on the postsynaptic neuron. But if they are clustered together like this, they can have a supralinear effect on postsynaptic neurons and drive the postsynaptic neuron strongly and robustly. And so previous people have postulated that perhaps learning takes advantage of a mechanism like this. So if learned information is clustered together on the dendritic branch, that can lead to a reliable activation of the neuron when those synapses are active. And that might lead to a reliable execution of the learned movements, a learned behavior. And we think we have evidence for that. So when these two synapses are co-active, they can have a strong impact on the postsynaptic neurons' activity. So now we became really interested in studying what happens when these synapses are co-active. And we find that almost all of these co-activity events happen during movement, during or right before movements. So we only see these synapses being co-active when the mouse is making the learned movement, okay? And yeah, what kind of movement are they making? Here is an example. 5 movement examples coinciding with the co-activity of a new synapse and the movement-related synapse next to it. And these are 5 different movements that don't coincide with the co-activity events. One is more reproducible than the other, and this is a highly reliable result. So when the new spine is co-active with nearby synapses, the resulting movement is much more similar to the learned movement pattern. So just to remind you, this is a learning of a task in which mouse is learning to generate a reproducible movement. And when the new synapses are co-active with nearby movement-related synapses, their movements tend to be the reliable, reproducible learned movement. So consistent with the idea, these coincident inputs to these new synapse-related clusters are driving these learned movements. All right, so these two synapses near each other are co-active. How does that happen? How are they co-active? Here's we can think of 2 simple scenarios. One is that they receive inputs from the same axon. Then you'd expect that their activity must be often co-active. So whenever the presynaptic neuron is firing an action potential, their downstream spines would be co-active as well, considering potential possible release failures, etc., which would reduce reproducibility. Perhaps a more exciting finding would be that these new synapses receive inputs from a different axon that is different from the nearby movement-related synapse. But somehow these different axons, different neurons, different presynaptic neurons show correlated activity, so that the postsynaptic spines would show correlated activity. This might be seen as an evidence that this synaptogenesis might bind new information streams onto this dendritic representation. Different axons, it's a new axon, nevertheless correlated with the pre-existing connection. This is a conceptually simple distinction, very hard to experimentally test, because when we are imaging this postsynaptic neurons, we're using sparse labeling. We cannot see anything beyond what is labeled. And so the only way we were able to think of to try to get at this question was to perform in vivo image longitudinally, identify these synaptic clusters, and then take out the tissue and perform 3D EM to visualize the tissue surrounding the image dendrite. You had a question?
Hi, Takaki. Let's see, we have a question from Zoom, which is from Dieter Jaeger. He asks, what are the presynaptic sources of coactive axons, thalamus, cortex, or other?
What is the source? Like where in the brain are they coming from?
Yes.
Okay, yeah, great question. We don't know. We're trying to look at that. We have a hypothesis, which is some of these likely come from the thalamus. So we think the thalamic inputs to the cortex sort of kickstart the population dynamics in M1, And at least some of these new synapses that form might be receiving inputs from the thalamus, but we have not shown that. Yeah, great question. Anything else? Okay. So, so we did that. We did perform the longitudinal in vivo imaging and took out the exact tissue that we were imaging and surrendered the tissue to this workflow of correlated electron microscopy, partnering with our local colleague Mark Ellisman's group. I'm not going to go into the technical details, but what we can do is the following. So we perform this in vivo imaging. We imaged this dendrite across learning and then took it out and performed a 3D EM. And we can manually identify the dendrite that we were imaging and reconstruct this. And it's very convincingly the same dendrite, and we can even identify the same synapse across preparations. And now we are able to zoom in and say, all right, this is a new synapse that formed right next to this pre-existing movement-related synapse. And we know that these two synapses show correlated activity. And when we reconstruct the presynaptic axons, even though these spines are only a micron apart from each other, we can show that they are connected with two different axons that run basically perpendicularly to each other. Okay. And this is almost always the case. It's not always, but almost always, these new synapses show functional clustering with other spines that receive different axonal inputs. Okay. So we think this is evidence for evidence that these new synapses are binding new information streams onto the dendritic representation. So with this and other lines of evidence that we that I don't have time to show you today, we propose the following model. So we start from a dendritic representation when we are trying to learn something new, and some of those synapses happen to be important for the learned behavior, right? And then those synapses are used and reinforced to be strengthened. They undergo synaptic potentiation. And the synaptic plasticity signal diffuses biochemically in the local neighborhood of the dendrite that induces filopodial protrusions. Basically, dendrites start searching for potential synaptic partners in the neighborhood. This process might be somewhat random, but if some of them happen to connect with axons that show activity that correlates with nearby synapses, then that connection preferentially gets maintained or stabilized so that they become stable new synapse. And this is a biologically feasible mechanism. So all of these, each of these aspects can be related to specific molecules, etc., that have been studied previously. And what is satisfying about this is that this is a simple, biologically feasible mechanism, but that achieves what we want to achieve during learning. So the result of this is that these synapses form a functional cluster that receive inputs that correlate, that receive correlated inputs. And as I said earlier, when nearby synapses become coactive, that reliably drives the postsynaptic neuron. And we think that might underlie this reliable activation of neurons that we see as a function of learning. So when the mouse is generating this learned movements, coincident inputs arrive to the functional clusters that have been expanded by these new spines. And that reliably drives the activity of these M1 neurons, leading to the reliable, reproducible population activity pattern. Okay. So that's the model that we propose. Yeah.
What do you mentioned? You think there's a You mentioned you think there's a biochemical signal from the strengthened synapse to kick off the formation. What do you think that is, and have you tested it?
We have not tested that directly, but actually, so that part of the work comes from this first author, Nathan Hedrick. He's a postdoc, excuse me. So he's a postdoc in my lab. His PhD work with Ryohei Yasuda showed a number of raw GTPases that can diffuse within 5-10 microns and change the plasticity threshold of nearby synapses. So we think something like that is doing what we see here. Yeah, good question. Any other questions before I move on to the second and hopefully a shorter part of the talk? All right.
I actually have a question.
Yeah, go ahead.
So this is the case for the nearby synapses, right? Like in the same dendritic compartments. But I was wondering if you've actually considered or looked into inputs from different compartments, like different compartmental dendrites, and then whether that time delays can actually initiate like a sequence of movements, sequence of motifs of movements.
Yeah, that's interesting. I'm trying to understand. So the sequence is across cells, right? So each cell is reliably active, and that follows. Some cells follow others, and we tend to think that those later neurons are driven by earlier neurons. So not necessarily the intracellular computation within the single neuron, but it's possible that maybe those later cells are driven with less coincident inputs, and so maybe their activation is delayed. And actually, that comes with an interesting observation, right? So these later cells are less reliable, less precise in their activity pattern. So your model could potentially explain that. So I like that. I haven't thought of it that way, but I think that's possible. Yeah.
We have another question from Zoom. This is from Simon Spomberg. He asks, if the animal is learning multiple movements, would you expect multiple types of correlated spines in the same neurons or different neurons underlying each of the movements? Both outcomes seem challenging because the first could lead to the growth and elimination of synapses could conflict. In the second case, we would seem to need a very large library of neurons.
Yeah, great question. I think it's both. So at the population level, the different movements activate overlapping but different sets of neurons. So in that sense, the plasticity is likely partially overlapping in terms of which cells undergo through this kind of plasticity. But even within individual neurons, there are mechanisms to compartmentalize this kind of processes. So, for example, different dendritic branches can compartmentalize this kind of mechanism. And indeed, Wenbyo Gan had a really nice paper a number of years ago that suggested that when a mouse learns to walk forward versus walk backward, the synaptogenesis that relates to forward versus backward are often found in different dendritic branches. So even in the same cells, there are ways to compartmentalize this kind of processes. Yeah. So yeah, great question. All right. So I'll go into the second part, switch gears and talk about this second project. And I should say this is a very new project ongoing. First time I'm talking about it in public, so it's going to be rough and preliminary. And I'm doing this looking for feedback. So yeah, I'm excited to hear what you think. So the big question is, okay, so we've shown that this population activity changes correlate with learning. So it happens during learning. And part of the activity happens before the movement. So it's definitely not just a result of the movement, but is it really driving the movement? Okay, so that's a question that we haven't been able to address until recently. Again, these things tend to be driven by technological development, and a technique that became available to us in the recent years is optogenetic stimulation with single cell resolution. Okay, so here we perform calcium imaging of a population of neurons, and then we can say, all right, I think these red neurons are interesting, so we'd like to activate them and use a special light modulator to selectively deliver light, a 2-photon light activation light to those cells and activate them very reliably. So these are the red neurons that are activated here. And notice that other cells are not activated at that time. And we can move the light to now activate these yellow neurons. And then these neurons are active. These are the yellow neurons, and other neurons, including the red neurons that I showed at the beginning, are not activated. And we can move to the third stimulation group here, the green groups of neurons, etc. So here we have the ability to identify neurons based on their activity because we're doing calcium imaging, and then selectively activate the neurons, a population of neurons that we think are interesting. And here, and we can do this in 3D. So we can image different planes and move across planes quickly and activate a subset of neurons in each plane semi-simultaneously. Okay, so this is the approach that we use to address this question. Is the activity in M1 able to drive movements? All right. And we use a very similar task. So here, the mouse is head fixed again. It has a lever. It hears a tone. So the task is to press this lever in response to a tone. And here's the mouse that's performing the task. So this left hand is pressing the lever forward, and it receives the water. So it's like you can also see the licking, right? And what we're going to do is, in a subset of trials, we're not going to play the cue. We're not going to play the tone to tell them to move. But instead, we activate some neurons in M1 using optogenetics and see what happens to the behavior. All right. So we trained mice with this task and imaged the activity of a population of neurons in M1 after 2 weeks of training. And reproducing our earlier results, we find reproducible activity patterns of these M1 cells. And some cells start right before movement onset, consistent with their role in driving the movement. So we identify those cells. And then in a small subset of trials, instead of delivering the sound cue, we activate those neurons, okay? And here's the result. So here again, I'll show you the same movie on the left. So this is a voluntary push. The mouse is pressing pressing the lever in response to a sound cue. We're not doing anything externally, or we're not doing anything to the brain. And here's another trial where we're not delivering the cue, but we're activating M1 neurons. And to the first approximation, I cannot tell the difference. All right, they're pressing the lever exactly how they would in the normal trial, except that there's no reason for them to press the lever. They're not hearing the cue, and also we don't want to reinforce them to press the lever in response to the opto stimulation. So we don't reward them in these trials. So this is a completely unreinforced behavior that is driven by the optogenetic stimulation. Yeah. And yeah, this is only like about 20 cells that are being activated by our stimulation, or directly by our stimulation.
Dieter asks whether these are still L2-3 neurons, and comments that they wouldn't project to the spinal cord.
That's right. Yeah, we are doing this all in layer 2-3, so they're not directly projecting down to the subcortical structures. That's right. Mm-hmm. Yeah.
For the recorded neurons you showed on the last slide, the neurons that fired the earliest looked pretty close to 0. And you said they were consistent with, maybe a premotorish delay. Would you expect that to be closer to like a few, 200 to 300 milliseconds before the onset of the movement? It looked like they're very close to 0 in the pseudo color plot that you showed.
Yeah, that's right. So in these trained animals, the reaction time can be like 100 to 150 milliseconds. And that's when these neurons start being active. Yeah.
I see. Okay. And in the case of the opto-induced push, what's the latency that you see between opto and the induction of the movement?
Yeah, good question. I think it's about the same, 100, 200 milliseconds, similar to the reaction time we see in the voluntary
Interesting.
trials.
So do you think if you moved to auditory cortex, for example, and stimulated neurons that were active during the tone, you would get a longer latency? Use the same Carmine preparation there?
Yeah, good question. Yeah, so at different levels of hierarchy, can we see the hierarchical processing? That would be the prediction, but I don't know if the delay is long enough to be able to result. But yeah, that would be definitely the prediction. I don't necessarily think this is auditory cortex dependent, but that's a separate
Right,
issue.
right.
Yeah, yeah.
Cool.
Yeah.
Have you stimulated neurons that you don't think are involved in the task?
I'll touch on that later. Yeah, we are just starting to do it. We're starting to analyze the results. Not much we can say at this point. Yeah, but I can say, so it doesn't have to be the right cell, right cells to generate some movements, but activating the right cells make better movements. Yeah, and I'll come back to the point. Yeah. Okay, so, and this was really surprising to me. I didn't think this should work. I didn't think the brain should be like, you just activate 20 cells and you generate the learned movement. I thought it was a more, distributed code across many cells, perhaps across different brain areas, right? But this is really showing that direct activation of 20 neurons is sufficient to induce this, maybe simple, but not more complicated than just a twitch kind of movement, right? So how is this happening? And we started looking at the movements more closely. So here is an analysis of voluntary trials. So, and we're looking at different trials, across trials, right? So and we're basically analyzing the quality of the movement. How similar is the movement that's induced in individual trials relative to what we call the learned movement? We identify consistent movements that are found across trials and see how well, how closely does the movement on individual trials resemble this target movement, what we call template here. In most trials, the movement is very similar. So this is an expert animal that is able to reproduce this movement reliably. And there are a small fraction of trials where the movement becomes dissimilar from the target movement. So, these are experts, they still make mistakes, right? The optogenetic stimulation-induced movements are less consistent. So here, grand mean shown in thick lines, and individual animals are shown in thin lines. So the really good learned movements are induced in something a little more than half of the trials of the optogenetic stimulation. And in other trials, the movement is still induced, but not quite the clean target movement, okay? So there's a variability, even though we're doing the exact same stimulation, right? But the resulting movement is variable from trial to trial. So we wanted to understand this.
Sorry, is that movement of the lever or?
This is the movement of the lever, yeah.
Okay.
Mm-hmm. Trial averaged, yeah. We're doing some analysis with the video as well. We're recording video. Yeah. But we think the lever movement captures a large fraction of the variance of movements. Other things correlate with that.
Yeah, hi. So, all right, sorry, this might be embedded in one of the earlier questions, but when you activate those 20 neurons, are they all activated simultaneously, or because they have some variability naturally? And I'm wondering if you activated them in a way that was consistent with the original activation, would the behavior be more consistent?
Yeah, I totally agree that that would be a prediction. We don't know. So right now we're activating all of them as simultaneously as possible. And it's just a short pulse of activation. We haven't played with the pattern, temporal pattern of that yet. Yeah. Good question. So, okay, so something is variable from trial to trial, although we are doing delivering the same light stimulation. So we wanted to understand this variability by looking at the population activity that is induced by this artificial stimulation. So we're activating 20 neurons, and at the same time, we're imaging all the neighborhood neurons, right, that we're not directly activating. And many of them are driven by this artificial stimulation indirectly, presumably through synaptic connections. And so here's a population activity pattern. This is actually from voluntary trials, averaged across trials. And you know, there are a lot of neurons, high dimensional. We reduce it using simple principal component analysis. And this we can reduce this population activity to a trajectory in the high dimensional space, or reduced dimensional space. Starting from before the movement here in an open circle, and the movement starts right here, and then the population activity transitions along this trajectory. And what happens when we're activating 20 neurons? So here's an average of optogenetic stimulation trials. So before the stimulation, the population might be around here, and then we stimulate right here, and then the population activity follows a trajectory. In this example, very similar to this natural trajectory, voluntary trajectory. And we find that this variability across animals of how well the learned movements is induced correlates very tightly with how well can we approximate the learned activity pattern. Okay, so here the x-axis is how close these two trajectories are. So the natural trajectory and the distance between the natural trajectory and the optogenetically induced trajectory, that's in x-axis. So closer these population activity trajectories are, the more similar the induced movements across trials. Okay. So it seems that this movement variability of induced movements seems to be related to the variability of induced population activity. Okay, and this seems so even on single trial level. Okay, so here's a single trial example trajectory, and actually this is a trial average, but so yeah, this is a natural trajectory, and now I'm going to show you 3 different trajectories of different groups of trials. Here is a group of trials where the induced movement is very similar to the template. Okay. And these are different groups of trials from the same animals where the induced movements are not very similar to the template learned movement. Okay. And you can see that when the resulting movement is very similar to the learned movement, the trajectory is also similar. But when the movements are dissimilar to the learned movement, the trajectories are also dissimilar. And this is a very high correlation on a trial by trial basis. Okay. Very significant correlation. So it appears that you know, even though we're applying the same stimulation to the same 20 neurons, the resulting population activity is variable from trial to trial and session to session. And that correlates with how closely we can reproduce the movement. But why is the induced activity different, even though we're applying the exact same stimulation? So this figure sort of shows a clue to that, right? So the optogenetic stimulation is applied at these field circles, right? But even before the stimulation is applied, the population is at different places in this population activity space. And in this quote unquote good trials, where the movement can approximate the learned movement, the pre-stimulation population activity is very close to the voluntary trials. But when this pre-stimulation state is far from this voluntary state, the induced activity is also very different from the learned movement. And so we analyze this pre-stimulation state, and we also see very tight relationship between how close the pre-stimulation state is to the template pre-movement state. That has a strong predictive information about how well the stimulation can induce the learned movements. So we're not controlling this pre-stimulation state, right? Different trials, the mouse is just in a different state. The M1 activity is in different states. But when where stimulation is applied, and when that happens to be when the mouse is M1 is in the say pre-movement state, right? And then this 20 cell stimulation is sufficient to generate the naturalistic activity trajectory and a very nice learned movement. Okay, but when the pre-existing state is far from this good preparatory state, let's say, their 20 cell stimulation is not sufficient to resemble the learned behavior. Yeah.
So this is if you're computing your principal components from all 100 neurons in this case. So if you restrict it to the neurons that you're stimulating, do you get is it the same story? So is it the preparatory state of the entire network, or is it just that the cells that you're stimulating, are quiet or primed and ready to go?
Okay, interesting. So okay. Yeah, good question. So yeah, I realize I didn't specify that. For all of this analysis, we're not including the cells we're stimulating. And we have a separate set of analysis that shows that direct stimulation is very reliable. So across trials, we don't see a clear variability, at least that correlates with the variability of the induced movements. Okay, so every time we're activating the 20 cells very reliably. But the response of other cells to that stimulation are variable.
I see. Yeah. And the pre-period is, I guess it's not necessarily super correlated between the 80 neurons that you used for this and the 20 neurons that you stimulate, since a lot of the variance seems to be explained by the pre-stim movement in like PC2 here.
Yeah, well, I'm not sure I understand your question there. Can you say a little more?
Yeah, so I guess what I'm wondering is A, what PC2 is, and B, these neurons are all just like activating during the movement. So in this case, you're saying that variability in the 20 neurons you stimulate in the pre-period does not explain much variability in the movement, but in these 80 other neurons it does. So the principal components, I'm just like curious what they're reflecting that could be different between those two populations.
Yeah, sorry, that's not what I said. What I said is that the response to optogenetic stimulation of the target cells is very reliable. Okay, so there's not much variability. We haven't done analysis to say which PCs corresponds to which kind of a variability, which cells, etc. Okay, yeah.
I was wondering if for your non-optogenetic trials, the ones that are just the tone-induced movement or
voluntary trials,
voluntary trials, whether or not this kind of like pre-tone activity then also correlates to the induced activity in the movements, or if there's something about the tone that actually is setting this state.
Yeah, thank you for that. The last part of your question is really interesting, and I don't know if I can address that, but that's exactly actually the next animation, the first part of your question at least. So yeah, so the question here is, is this something really unique to optogenetic stimulation? Just somehow the state responds differently, the population responds differently to the same optogenetic stimulation, or are we actually uncovering something that we see in the natural behavior, right? So that drove us to do an analogous analysis to see if the pre-movement activity state can predict the voluntary movement quality, right? And so that's the last data slide, data that I wanted to share with you today. And the short answer is it does. So we can look at the where the population activity is during the state right before the Q comes on. And we can do a fairly good job predicting how well how good the trial is going to be, right? So if the so there is a specific, so maybe I can go to the schematic. So this is what we think is happening here. So there is a preferred preparatory state of the population activity, and the mouse often in most trials can enter this state in experts, and at that point, the involuntary trials, it generates a reproducible trajectory that is reliably paired with the learned movement. If the network state happens to be in this state, activation of 20 M1 layer 2-3 cells is sufficient to mimic this population activity, okay? But this preparatory state can be variable. And in some trials, you're over here. And then whether you're stimulating them artificially or the mouse is induced to move by a sound cue, the resulting activity is not what the population is trying to achieve. And then the induced movement is also very different. It's the same thing. If you happen to be over here, you're in the wrong state, the stimulation or cue will result in different activity patterns leading to different movements. So there's something that is really setting the preparatory state so that the population is really ready to drive this learned movement activity pattern. And that's when, even in natural environment, in the natural trials, the mouse is able to generate the right movement. And in those trials, we can stimulate 20 cells and generate the same movement, essentially. Yeah. So, yeah.
How would you quantify the brain state? Like, are you doing like spectral analysis looking at the or that?
Yeah, what do I mean by state? So it's just a location in the population activity space. So this is all based on single cell activity from, on average, about 400 neurons. And so basically it's a 400 dimensional space, right? Every cell has an activity level, and it's a 400 dimensional space. We reduce it just to reduce noise, but that's not very important. So basically, so that's basically which neurons are active at the time. Right, so that's what we mean by state. So when the right neurons are slightly active, that's the right preparatory state. Yeah.
So it seems like the measurement of the so it seems like the measurement of movement, how its similarity to the template, like as you show, is like the distance to the template. So have you looked at the other aspect of the kinematics, like speed, like acceleration, or the extent of the movement, like really how much it overshoot or undershoot, and how whether that would be related to what internal state, the optogenetic stimulation?
Yeah, excellent question. We need to do a lot more. This is really new, and we haven't done a whole lot. One thing I can tell you is that optogenetically induced movements tend to be smaller in amplitude. So compared to voluntary trials, they don't push as far. So yeah, that is not something we're mimicking quite well, and we don't know why. But yeah, there are other aspects that we should look at as well. Yeah.
Chris Rogers asks, is the pre-movement activity state changing slowly, perhaps reflecting arousal or motivation? Or is it changing rapidly, perhaps reflecting movement preparation? And could neuromodulators set the preparatory state?
Yeah. These are all the questions that we think about. We don't have a really good answer to. How quickly are they changing? We don't know. We don't know. We only did trial by trial analysis. Within trials, can they rapidly enter the right preparatory state? We don't know. We don't know. I imagine so, but we don't know. Is it neuromodulator driven? Very likely. Although we think it has to be a high dimensional signal, or at least it's not a single dimensional signal, because you must be able to prepare different movements, right? So it's a little hard to imagine that a single neuromodulator is sufficient for that. But yeah, we don't know. This is all speculation. Yeah, great question. We're getting that. We're really interested in the function of noradrenaline in this aspect as well, because there's a really nice paper from another group that showed that noradrenaline can really flip the state between quiescence and movements in M1. And we're interested in looking at that. Yeah.
So it seems like a component of learning might also be getting in the right preparatory state.
Mm-hmm.
So in the first part, you showed the learning is really about the learned movement, but I'm wondering if you've thought about learning of the preparatory state.
Absolutely, I agree. I agree with you. I think I think it's both, right? So even if we can somehow force the system to be in the right preparatory state in a naive animal, I don't think stimulation can generate the whole activity. So this whole activity trajectory, I think, is also learned by synaptic plasticity. So I think both have to be learned, right? Yeah. I know less about how they're learning the preparatory state. Even less about, I should say. Yeah.
I just have a clarification question. I might have just missed it from your last slide, but so in the naturalistic voluntary movement, did you see the trend that the neural activity was set at the correct initial state most of the time, or?
They're more likely to be. So it's this analysis. So basically where, yeah, a lot of the trials lie in this regime where the preparatory state is very close to the target, and then their movements are good. But in some trials, they're far from that right target, and then their movements are also bad. Yeah. All right, so just to summarize the second part of my talk, we think we showed that targeted stimulation of 20 movement-related M1 neurons is sufficient to induce naturalistic movements, but that doesn't always happen very successfully, and variability of induced population activity reflects the variability of induced movements. And the pre-stimulation population activity state has predictive information about what kind of population activity and movements are induced by optogenetic stimulation. Okay, so and with that, yeah, I'd like to conclude by thanking all the lab members. The first part of the talk is the postdoc work of Nathan Hedrick, very talented individual who will be on the job market this year. So keep an eye on him. And the second part is run by An Wu, as another postdoc, extremely, one of the best experimentalists I've ever seen that makes very difficult experiments easy. So yeah, I can't emphasize how hard it is, the experiment that we showed at the beginning. But yeah, An makes it look easy. And yeah, I'd like to thank all the other members and thank you all for your attention. Yeah, thank you. Happy to keep answering questions, but I don't want to keep everyone from your next engagement either.
Is there any question from the audience?
We have one question on Zoom from Dieter Jaeger. He's going to try to unmute himself and ask it.
Yes, hello, am I audible?
Yes, you are. It's kind of soft.
Okay, so wonderful talk. Thank you very much. That was great.
Thank you.
I have a question for the first part. Since there's dopamine innervation of motor cortex, I was wondering whether the spine plasticity was dependent on dopamine release.
Yeah, good question. We actually have looked at that, and I know there's a bunch of studies that have looked at more dopamine's role in regulating motor cortex plasticity. In our hands, we see very little dopaminergic axons in the mouse M1. So we didn't follow up on that. Yeah, I don't know why. Actually, I haven't seen a lot of convincing anatomical evidence that midbrain dopaminergic neurons project to M1. So there could be other sources of dopamine. But so we're looking more at other neuromodulators. For example, we recently had a paper showing that the cholinergic innervation of M1 is important for regulating inhibitory neurons that regulate excitatory plasticity. So there's that kind of a neuromodulatory control of learning-induced plasticity in M1.
Right, thanks a lot. Yeah, and I agree that the dopamine has been shown more in premotor cortex and ALM, for instance. I guess that leads to the question, how much of the motor learning you think might also happen, say, in M2 and higher order motor cortices?
Yeah, good question. I think it's distributed. One thing I can say is that when we block synaptic plasticity in M1, we can block learning. So it seems essential. But that doesn't mean that this is the only place where changes are taking place. I don't think it is.
Thank you.
Thank you.
Thanks.
Thank you.